Hi friends. Today we are going to be talking about this picture. It is showing two tomatoes, and on the picture it says, what is the difference? So I want you to start thinking about what could have naturally caused these two tomatoes to be different, and then also how could humans have intervened to cause these two tomatoes to be different? You are going to need two materials for this video today. First one is your science journal, and the second one is something to write with, preferably a pencil. All right, first task I want you to do is to write down the learning objective. So our learning objective, so by the end of this video, you are going to be able to explain the multiple technologies that humans can use to affect traits in organisms. Please go ahead and pause the video, write this down in your journal, and then click play when you are done. Okay, so now that you have that written in your journal, I want you to look at the picture. And I have chosen to use dogs as an example today for at least one of our technologies because dogs, if you don't know, are all the same species, which is why you're able to breed lots of different species of dogs, lots of different types of dogs together because they're all the same species. So our first thing that we're going to talk about, make myself a little bit smaller. First thing we're going to talk about is selective breeding. You don't need to write anything down yet, but selective breeding is the process in which humans decide what traits are important. They choose parent organisms that show these desired traits. They then select the best offspring to breed for the next generation. And then number 4, repeat the process until there is a population of organisms with the desired traits. So if we look over here at our example, you can see the first generation in these dogs. Most of them are pure white, and then we have a few of them that have spots. So if you were trying to get dogs that have spots or more spots, you would choose 2 parents that have at least some spots, right? And so that is shown here by these 2 red check marks. Moving on to the second generation, so we're showing it could be a couple of different families. It could be just one litter from these 2 dogs specifically. But we see we have now several dogs with at least some spots, but we have a few dogs, so 2 here, that have even more spots, right? And if our goal is to get dogs with more spots, we're just going to keep on picking the parents that show these traits. So then by our third generation, now we're having dogs with more spots overall. And if we wanted to keep on going, again, you would just keep on choosing the dogs with the most spots and have them breed for the next generation. Okay, and so humans, this is super easy to do. Some downsides of it is it's going to take a really long time, especially with depending on how long it takes for animals to reproduce. Some of them, it only happens several or like one time a year. Sometimes it's several times a year. You really have to think about the animal, or more often than not, we're probably doing this with crops. So think about growing seasons and how often, or like what climate you're able to grow certain organisms in. My second example to show you, which we might be a little bit more familiar with here in Iowa, is so let's say a dairy farmer may select from a herd of larger bulls and breed them with cows that have the best milk production, right? Because if you're a dairy farmer, you want your cows to produce a lot of milk, right? So it's probably going to be your larger cows, but then also that could be a second quality. So in order to do, or so by doing this, this would give the farmer the best dairy cattle population. So again, just another example, you're choosing the animals with the qualities that you would like, and you choose to purposefully breed them, right? Selective breeding. All right, our second example then is called genetic engineering. So this is a little more scientific. So genetic engineering is when humans transfer a desired gene into another organism's set of genes. This is much faster, right? We don't have to wait for, for example, a dog, we don't have to wait for it to produce like several different litters of puppies, right? You could just take the gene. Now this does have to happen like when it's an embryo, right? It can't be a developed organism. So you can't take like our example here is with apples or tomatoes. If you want to think of our original one, you can't take an already tomato and put a gene into it, right? It has to be like at that seed, like at a very, very, very early in replication. This is a sophisticated version of cut and paste, right? Or copy and paste if you want to think about it as that like we use on like our Google Docs. And so what they do here is, in this example, we have a bacterium with insect resistance gene. And we actually do this in a lot of our foods. You might hear them called GMOs. I know you guys have finished reading The Omnivore's Dilemma, and so you're probably going to see a lot of connections here. So they're going to take this gene that is very specific to insect resistance. They're going to place that in the fruit. In this case, it's going to be an apple. And that process is called genetic modification. It's obviously it's a lot more detailed than what I'm explaining, but right now I just need you to understand the difference between these two processes. So they're going to put that gene into like the seed of a fruit, and then hopefully that apple then is going to grow with that insect resistant fruit. I say hopefully because like we know with science, it's a lot of trial and error. It doesn't mean it's specifically that gene is going to be expressed. And so in order for this to happen, they have been tested and tested and tested many, many times. A second example of this would be our corn here in Iowa. So in Iowa, it is important to produce corn that is resistant to insects so that the insects do not eat it all. Bioengineers have created BT corn, so that is like the #1 corn that's produced here in Iowa and as our nation as a whole. And so bioengineers have created BT corn by inserting a gene from the bacteria. Not even going to pronounce it because that is in Latin. But you can see it's short for BT. And then they put that into the corn, right? And so if you are a farmer, you live near a farmer, you might, or maybe even just from reading The Omnivore's Dilemma, you might have heard of BT corn before. Okay, so now what I want you to do is you are going to draw a model. So in your journal underneath where you put that learning target, and you can go ahead and pause the video and you can go back and look at any of the other slides, or you can use anything we've said in the video, but I want you to label one of the circles as selective breeding and the other one as genetic engineering. And then we're picking out similarities and differences. So anything that selective breeding and genetic engineering have in common, we're going to put in the middle, and then their differences put on either side. Go ahead and pause the video. I would like you to have at least 2 things in each of the 3 sections. Go ahead and pause the video, and then you can play it when you are ready. Okay, so your final task is we're going to return to that original photo. Remember, these are our 2 tomatoes. And so I want you to think about which process that you have explored in this task do you think is probably responsible for the larger tomato? I want you to pick one of the ways humans intervene with organisms and draw a flow chart. Remember, so that means like step 1, they do this, step 2, they do this, step 3. So draw a flow chart describing the process that may have created the larger tomato. So you can pick either one, whatever you think is probably a better explanation for this photo. You can go ahead and put that in your journal, either underneath your Venn diagram that you just made your model, or you can put it on the next page.